Hey everyone, welcome back to MedBite, the channel where we break down complex medical topics into bite-sized pieces. Today I have an exciting topic lined up for you: immunology. Immunology is a fascinating field that explores the intricate workings of our immune system, and in this video, hopefully, you will discover how our immune system protects us from diseases, infections, and much more. Before diving headfirst into this subject, I would like to clarify some fundamental concepts which I'm going to begin explaining in the next slide. The general immune system is made up of two subdivisions, the innate immune system and the adaptive immune system. We will discuss more about the distinction between these two immune systems later on, but for now, just appreciate that these are the two distinct arms of our general immune system. So here is a visual illustration of the two arms of the general immune system. An important constituent of the general immune system are white blood cells, also known as leukocytes. Now, that's not to say that the immune system is only made up of leukocytes. There are certain non-leukocyte cells, but by and large, the predominant cell type in the immune system are leukocytes. So some leukocytes play an active role in the innate immune system, others play an active role in the adaptive immune system, and some play a key role in either. There are a few broad categories of leukocytes, as written here: neutrophils, basophils, eosinophils, mast cells, lymphocytes, and monocytes. Lymphocytes are also known as lymphoid cells, so just keep that in mind. Neutrophils, eosinophils, basophils, and mast cells are collectively known as granulocytes, whilst the remaining two categories, lymphocytes and monocytes, are known as agranulocytes. The roles of each cell type will be discussed in future videos, and for some of these cell types later on in this video as well. Now let's shift our focus onto monocytes. There are two subsets of monocytes as I have displayed here: dendritic cells and macrophages. Now it's important to understand that firstly, monocytes themselves are found in the blood, whereas dendritic cells and macrophages are both found in the tissues. And that's why monocytes are quite mobile. They can move quite quickly from one part of the body to another, whereas macrophages and dendritic cells are confined to the region they're in. Another important thing to understand is that monocytes don't have to differentiate to form dendritic cells and macrophages. In fact, a lot of monocytes remain in their undifferentiated form while circulating in the bloodstream. I will talk more about the roles of dendritic cells and macrophages very soon, but not yet. Now let's look a bit more at lymphocytes, which is the other type of agranulocyte. As I've illustrated here, there are three subtypes of lymphocytes. You have B lymphocytes, also known as B cells, T lymphocytes, also known as T cells, and natural killer cells. As mentioned before, I will discuss the roles of each leukocyte in the future, but just to provide an important distinction between B cells and T cells. The reason why B lymphocytes are called B lymphocytes and T lymphocytes are called T lymphocytes is because of the location where they mature. You see, B cells and T cells are both initially produced in the bone marrow, but B cells undergo maturation in the bone marrow as well, whilst T cells migrate from the bone marrow to the thymus gland, where they mature over there. So because B cells mature in the bone marrow, they're called B cells, B for bone marrow, and because T cells mature in the thymus gland, they're called T cells, T for thymus. And then you obviously have natural killer cells, which do play an important part in the innate immune system. Continuing on, I would like to define another very important term in immunology: a phagocyte. A phagocyte is a term used to describe any cell within the immune system that specializes in engulfing and destroying foreign particles, for example, pathogens, cellular debris, and other harmful substances. So here is the definition of a phagocyte written out for you. There are many cells which do exhibit phagocytic properties, but the main phagocytes that exist are neutrophils, macrophages, and dendritic cells. Now, if you remember, macrophages and dendritic cells are both derivatives or descendants of the monocyte. Neutrophils are found circulating in the blood, whereas macrophages and dendritic cells, as I explicitly mentioned earlier, are found in the tissues, where they are largely confined to a specific region. Now, just to reiterate, these three are the main phagocytes, but there are other cells which do exhibit phagocytic properties. For example, monocytes, which haven't differentiated into a dendritic cell or a macrophage, also do exhibit phagocytic properties, but not to the same degree as the three phagocytes I have listed below. One final thing I would like to discuss here are antigens. Antigens are very important. They're an important term that will be coming up time and time again whilst I talk about immunology, so it's important that you have a base definition for this term. An antigen is a substance that is capable of triggering an immune response. Antigens are found on all pathogens and all cells, including viruses, bacteria, fungi, toxins, and even the cells in our body, which are known as self cells. It's important to understand here that antigens have the capability of triggering an immune response, but they don't always lead to an immune response being triggered. What I mean by this is, yes, of course, an antigen on a virus or on a bacteria will lead to an immune response being triggered against that pathogen. But our cells, or the cells in our body, the self cells, they also contain antigens too. But these antigens, most of the time, won't trigger an immune response, because if immune responses were to start being triggered against self cells, this would lead to a lot of disease, autoimmunity, and destruction of healthy tissue, which we obviously don't want. Antigens are typically proteins found on the surface of pathogens and cells, but they can also be found intracellularly. Good. Now that we have clarified some of the fundamental concepts in immunology, let's discuss the role of the immune system in more detail. It's very difficult for me to just state the role of the complex immune system in a single sentence, and that's why I'm going to use the concept of the 3 R's: recognition, response, and regulation. All of the roles of the immune system, or the roles that the immune system fulfills, can be boiled down to one of these 3 features: recognition, response, and regulation. So let's first talk about recognition. Recognition refers to the immune system detecting or recognizing an antigen. Logically speaking, this is very important because let's say our body were to be infected with a viral agent, our immune system needs to recognize, it needs to acknowledge that the body has been infected. If not, then there are going to be problems. Now, as mentioned earlier, there are 2 arms of the immune system: the innate immune system and the adaptive immune system. And both of these immune systems go about recognition in a different manner. The innate immune system contains various immune cells. These immune cells are all equipped with a variety of pattern recognition receptors. These pattern recognition receptors look for molecular patterns shared by multiple pathogens. These are known as pathogen-associated molecular patterns, abbreviated to PAMPs for short. Let me give you an example. We know that most bacterium contain a cell wall, and in bacterium, this cell wall is made up of peptidoglycan. So in the innate immune system, the cells with their pattern recognition receptors can look for peptidoglycan, since this is a shared feature of most bacterial pathogens. So in this example here, your pathogen-associated molecular pattern is peptidoglycan because it is associated with the bacterial pathogen. It is a pattern found across most bacteria. Now let's talk about the adaptive immune system. The adaptive immune system largely involves 2 types of leukocytes, or 2 subtypes of leukocytes: B lymphocytes and T lymphocytes. B cells produce a special protein, which I'm sure you would have heard of, called antibodies, whilst T cells contain receptors bound to their surface, known as T cell receptors. Antibodies and T cell receptors both look for antigens that are unique to a specific strain of pathogen. What I mean by this is, taking the previous example, we discussed that across most bacteria, a prominent pathogen-associated molecular pattern is peptidoglycan. But antibodies and T cell receptors would not look for antigens like peptidoglycan because this antigen is not unique to a certain strain or species of bacteria. It is too general. Instead, the adaptive immune system would look for antigens that are found only on a specific species or strain of bacteria. And that's why the adaptive immune system is described as a highly specific immune system, whereas the innate immune system is described largely as a general immune system. Another small little bit I'd like to add, just for the record, is that pathogen-associated molecular patterns are still antigens, because remember what I said about antigens. They are substances that are capable of triggering an immune response. And in the context of the innate immune system, your PAMPs are certainly capable of triggering an immune response if detected by the innate immune system. Moving on to the next R, we have response. So once the immune system discovers that there is a foreign agent in the body, once it is past the recognition stage, it needs to mount an immune response against it. So there are 2 types of immune responses: cellular, in which the response is mediated by cells, and humoral, in which the response is mediated by molecules instead of cells. 2 very prominent cellular responses launched by the immune system are phagocytosis and contact-dependent killing. So in phagocytosis, quite simply put, a phagocyte, which I discussed earlier, engulfs a pathogen, it brings the pathogen inside of itself, and inside of the phagocyte, the pathogen is then systematically destroyed and broken down into smaller constituents. In contact-dependent killing, a subset of T cells, known as cytotoxic T cells, more on this later in future videos, and natural killer cells, destroys a self cell infected with a pathogen. So there's a slight difference between the two that I would like to explicitly state. Phagocytosis is useful for destroying a pathogen itself, so it's useful for extracellular pathogens. But once a pathogen enters a cell in our body, then contact-dependent killing is the preferred mode of destroying that pathogen, because phagocytosis involves the destruction of the pathogen itself, whereas contact-dependent killing involves the destruction of a self cell that is infected with the pathogen that the immune system is trying to destroy. So let's talk a bit about humoral responses. So a humoral response is one that is mediated by molecules instead of cells, so in stark contrast to a cellular response. The two prominent humoral responses launched by the immune system are antibodies and complement systems. So let's first talk about antibodies. Antibodies are special proteins that are produced by B cells, as I mentioned earlier. Antibodies are also commonly referred to as immunoglobulins or immunoglobins, so do be aware of these synonyms. So how do antibodies actually function? Well, antibodies bind to the antigen on the surface of their target. The target might be a pathogen, it might be a cancer cell, it might be a toxin. The antibodies bind to the antigen on the surface of the target, and it has many effects, or it can cause many effects, some of which I will list on the next slide. So firstly, neutralization can occur. So when the antibody binds to the antigen on the pathogen, for example, it will prevent that pathogen from infecting other body cells. So it kind of acts as a kind of containment measure. Next, you have opsonization. So opsonization refers to the process by which the antibody binds to the antigen on the pathogen and then subsequently alerts nearby phagocytes to the location of the pathogen. So it acts as a signaling molecule. In this example, the antibody is acting as an opsonin, hence the name of the process, opsonization. Another very important mechanism of antibodies is antibody-dependent cellular cytotoxicity, abbreviated to ADCC. So here I've drawn a little bit of a picture just to give you a visual illustration of how this works. So let's say that we have a pathogen, or we have a cancer cell or a toxin. It can be anything. But the point is that your antibody will bind to the pathogen, and the antibody will also simultaneously bind to a natural killer cell. So what it does is it's essentially bringing the pathogen and the natural killer cell together in close proximity with one another. The natural killer cell will then proceed to destroy the pathogen. So the role that the antibody is playing here is it's enabling destruction of the pathogen by bringing together a very powerful member of the immune system with the target that needs to be destroyed. Another very important humoral response, which I mentioned earlier, was the complement cascade, also known as the complement system. So over here, what happens is that a pathogen activates a protein that is a part of the complement system. This protein then activates another protein. This protein then activates another protein, and so on. And so that's why we call this a cascade, because it's a cascade of reactions. This leads to the formation of various end products, which all help to destroy or disable the pathogen that initiated this complement cascade. More will be discussed on this concept later, that's why I've left this slide blank, but it's just important to be aware that this is one of the body's or the immune system's key humoral responses. Moving on to the final R, we have regulation. I want you to think of the immune system as a weapon. It's very useful against foreign entities like pathogens, cancer cells, toxins, but if we're not careful, then this weapon can start damaging healthy tissues in our body. So regulation aims to ensure that the immune system is carefully monitored and it is prevented from damaging our own self tissues. There are multiple systems in place to implement strict regulation, some of which I will discuss right now. One of the key regulatory mechanisms in place is multi-step activation. The initiation of an immune response requires numerous steps to occur, and what this does is that this minimizes the risk of inadvertent, i.e. accidental activation of an immune response. Let's say, for example, by accident, a couple of steps were to be fulfilled in the pathway to launching an immune response. Since there are many steps involved in launching an immune response, this would eventually lead to the immune response not being initiated. But let's say that the immune response only required a couple of steps to be fulfilled. This would increase the likelihood of an inadvertent immune response from being initiated or being initiated. Another very important regulatory mechanism is the presence of T regulatory cells, also known as Tregs. T regulatory cells regulates the activity of other T cells in order to maintain immune tolerance to self-antigens. Now, immune tolerance basically refers to the fact that the immune system is not going to recognize a certain antigen as foreign. So by maintaining immune tolerance to a self-antigen, what this means is that no immune response will be launched to the antigens that are being expressed by the local native cells of our body. And this is, of course, a good thing, because if the immune system were to start responding to the self-antigens, then we would have autoimmunity. And another very important regulatory mechanism is checkpoint proteins. So checkpoint proteins are proteins that are released by self cells, so cells that are naturally found in our body. And these self cells release checkpoint proteins to prevent the immune system from attacking it. A very common checkpoint protein is the PD-L1 protein, which is released by many of our own body cells. This PD-L1 protein binds to a receptor on the T cell known as the PD-1 receptor. What this does is that it prevents the T cells from attacking the cell which released the PD-L1 protein. So this is a very good mechanism of ensuring that the T cells, at least, don't attack the self cells. Unfortunately, cancer cells also express or release a lot of PD-L1 proteins, and in that way they avoid the immune system, or they avoid the T cells of the immune system. So that is definitely a problem. Anyways, that was it. We have covered the 3 roles of the immune system, the 3 R's. There's just one final thing I'd like to talk about, and that is to distinguish the innate and adaptive immune system a little bit more. I'd like to just outline some of the key differences. Firstly, I'd like to talk about the difference between the specificity of response. I mentioned this earlier, but the adaptive immune system looks for specific antigens, whereas the innate immune system looks for non-specific antigens, which are found across a broad range of pathogens. And these non-specific antigens we called pathogen-associated molecular patterns. So that is why the innate immune system's response is relatively identical to pathogens belonging to a broad group. For example, the innate immune system would respond in a very similar or almost identical way to various species of bacteria, whereas the adaptive immune system, which looks for very specific antigens, would have a varied response to different species of bacterium. Even if they belong to the same genus or class, there would be a slight difference, or rather a noticeable difference, between how it goes about responding to the pathogen. Another key difference between both immune systems is the development and speed of the response that either immune system launches against the foreign agent. The innate immune system launches an immune response against a pathogen or a foreign agent very quickly, but this immune response is short-lived, whereas the adaptive immune system takes a bit longer to launch the immune response, but once it does, the immune response is long-lived. Another key difference between both immune systems is the presence of immunological memory. The innate immune system does not possess immunological memory. So what this means is, let's say that I were to be infected by a specific strain of virus twice, both 2 months apart. The innate immune response would remain relatively constant both times and would not improve even despite repeated exposures to the same strain of pathogen. But this is in contrast to the adaptive immune system, which does contain immunological memory. So what happens here is, the first time I'm infected by a virus, the immune response will take its time. It will build up the necessary resources to attack and destroy this pathogen. But if I'm infected by the same strain of pathogen again, the adaptive immune response would be better, because the adaptive immune system would remember the specific pathogen and enable a faster and more efficient immune response, and thus the period of infection would be significantly shorter. And one final key difference between both immune systems that I would like to highlight are the constituents of either immune system. The adaptive immune system is largely made up of B cells and T cells and antibodies which are produced by the B cells, whereas your innate immune system is made up of quite a diverse range of cells and macromolecules. Physical barriers, for example, the ones found in the skin and the mucosal membranes, form a key part of the innate immune system. Phagocytes are also integral to the innate immune response, and as I mentioned earlier, the main phagocytes that I would like you to know are dendritic cells, DCs, macrophages, and neutrophils. The first 2 are derivatives of monocytes. The third is a direct type of leukocyte. You also have natural killer cells, which are very important to the innate immune system. The complement system, which we briefly discussed, but I will talk more about in detail in future videos, and granulocytes, which refers to a select few leukocytes, namely mast cells, basophils, eosinophils, and neutrophils. So that's all for today. Hopefully, at the end of this video, you feel that you have some kind of understanding of the basics of immunology. I will be making more videos that will explain some of the concepts that I've talked about in greater detail. There is still a lot more to cover, but this should at least set you for a good foundation and put you in the right platform to understand immunology in a structured manner. Thank you very much for listening. This is MedBite here. And if you like this video, then please stay tuned for more videos that I put out soon, hopefully. Thank you very much. Thank you.